Hello everyone. Today we will be talking about rotavirus. Our focus question is why did the mechanism of the first rotavirus vaccine research that it failed, and what is the modern mechanism? This is a background slide. Coming up to what is the viral structure or organization, rotavirus named after rota, which is Latin for wheel, because under the EM microscope it looks like a wheel. It is a non-enveloped Viridae member with three concentric protein layers. Outer layer is VP7 plus VP4 spikes. Those handle entry and define the G and P strain types. Middle layer is VP6, which sets the group A through H, and group A is the one which causes the most amount of disease. Inner core is VP2 plus VP1 polymerase and VP3 captain enzyme. The genome is 11 segments of double-stranded RNA. Each segment codes for one protein, and the whole viron is about 75 to 95 nanometers. There are four important cell entry mechanisms. The first is binding. VP8 star grabs sialic acid or histoblood group antigens, depending on strain. Second is co-receptors, which important ones are integrin and HSC70 in liver drafts. Third is uptake, which is notably not done by clathrin or endocytosis, but rather lipid draft endocytosis. Fourth is uncoating. The low endosomal pH knocks off VP7 and VP4 disrupts the membrane, releasing the double layered particle, the DLP, into the cytoplasm. That DLP is transcriptionally active and starts making mRNA immediately. The next slide, we discuss the replication and assembly, which is basically all replication happens inside the viroplasm. These are membrane-free, phase-separated droplets organized by NSP2 and NSP5, visible about 2 hours post-infection. Inside the VP1 makes positive strand RNA, which gets packaged into a new core, and then the negative strand is made inside the shell. So the double-stranded RNA is hidden from immune sensors the moment it exists. Assembly goes core, then DLP with the VP6 layer, and ER budding mediated by NSP4, and finally the triple layered particle gets VP7 and VP4 added. The diagram shows the whole cycle. Coming up to what is the innate immune antagonism, rotavirus actively shuts down immunity, and the main player is NSP1, which is a E3 ubiquitin ligase, meaning it targets the host protein for degradation. It degrades IRF3, 5, and 7, and blocks interferon production. It degrades B TRCP to block NF-kappa B and shuts down inflammatory cytokines. On top of that, viroplasms keep DS double-stranded RNA sensors like TLR3 and RIG1 and NSP4 leaks calcium from ER to disrupt signaling further. This diagram shows how the ER and how the rotavirus blocks the immune system. The most important slide is how does the rotavirus cause disease? The main problem is obviously diarrhea. Three routes converge on diarrhea. First is secretory. NSP4 is an enterotoxin that opens the CRF3 and calcium-dependent chloride channel, dumping chloride and water into the lumen. This motility, enteric nervous system, and vagal signaling speed of gut motility. Finally, osmosis, cell lysis kill enterocytes, reducing enzyme expression and absorptive areas, so the nutrients and water stay in the gut. All three together cause severe dehydrating diarrhea, which is prototypical of this disease, and that is why retrovirus is the leading killer of young children, and why vaccines are very important. We're going on to the next slide.
Hello, my name is Travis, and I'll be talking briefly about RotaShield. Before modern rotavirus vaccines were available, rotavirus is one of the leading causes of severe diarrhea in infants and young children worldwide. To address this problem, RotaShield became the first vaccine developed specifically to prevent severe rotavirus gastroenteritis. RotaShield was approved in the United States in 1998 and was developed by the pharmaceutical company Wyeth, which was later acquired by Pfizer. One important feature of this vaccine was that it was administered orally rather than by injection, making it easier to give to infants. The vaccine was designed to reduce severe diarrhea, dehydration, and overall hospitalizations caused by rotavirus infection. Infants received the vaccine in a 3-dose schedule at approximately 2, 4, and 6 months of age. RotaShield was a live attenuated vaccine, meaning it contained weakened viruses that could stimulate the immune system without causing severe disease. It was also a tetravalent vaccine, which means it targeted 4 different rotavirus serotypes as shown in the figure on the left. To achieve this, researchers created the vaccine using rhesus monkey rotavirus strains combined with human reassortant strains. After being taken orally, the vaccine entered the intestinal tract where rotavirus normally infects the body. The weakened viruses mimicked a natural infection and stimulated an immune response in the gut. This led to the production of protective antibodies, particularly IgA antibodies, which help defend against future rotavirus infections. Although RotaShield was effective at preventing rotavirus disease, Concerns arose shortly after its introduction. Researchers found reports linking the vaccine to intussusception, which is a rare type of bowel obstruction where one section of the intestine folds into another. The risk appeared to be highest within 1 to 2 weeks after the first vaccine dose. Studies estimated that there is approximately one case of intussusception for every 10,000 vaccinated infants. Because of the safety concern, the CDC suspended its recommendation for Rotashield in 1999 while investigations were conducted. Later that year, the manufacturer voluntarily withdrew the vaccine from the US market. This decision highlighted the importance of balancing vaccine benefits with potential risks. Even though Rotashield was withdrawn, it had a lasting impact on vaccine development. The experience demonstrated the importance of post-marketing vaccine safety monitoring, which involves continuing to evaluate vaccine safety after approval and widespread use. Lessons learned from Rotashield led to stricter safety testing and larger clinical trials for future rotavirus vaccines. This work ultimately contributed to the development of newer vaccines, including Rototec and Rotarix, which will be discussed later in the presentation. Today, these vaccines continue to be monitored for rare adverse effects such as intussusception, helping ensure that their benefits greatly outweigh their risks. Overall, while Rotashield had a relatively short time on the market, it played a major role in advancing rotavirus vaccine research and improving vaccine safety standards.
And so to replace Rotashield, Rototec was introduced in 2006. It's an oral vaccine given to infants starting from when they're 6 weeks old. You need 3 doses for completion, and it's required to complete it before 32 weeks of age. And so they recommend that you get the doses between 4 and 10 weeks apart. And so this is a live oral vaccine that takes advantage of a reassorted W3 bovine virus, which is a cow rotavirus, non-infectious to people, and it's been engineered to express either G1, G2, G3, G4, or P8 human glycoproteins. And the studies on this show that it does drive production of CD4 memory T cells, showing that it activates the adaptive immune response. And so here's just some data on the on the vaccine. We can see on the left, it's comparing seropositive to seronegative. Essentially, the dark blue bars represent individuals who had antibodies in their plasma after the vaccine, and the light blue is the placebo group, and we see that getting the vaccine does stimulate antibody production. And then on the right, we see that getting the vaccine greatly reduces the number of severe rotavirus cases. So that doesn't necessarily prove that it stops infection, but it does stop severe symptoms, which is good. And it shows that it's effective. However, there are some side effects like vomiting, diarrhea, stomach pain, bloody stool, and intussusception. Intussusception is pretty unique. It's where the small intestine slides into the adjacent segments. It's pretty serious. Some symptoms are like severe stomach pain. It can lead to cutting off the blood flow to the area, tissue death. It's really serious and it needs to be treated immediately, but it is also pretty rare.
Hi, my name is Leah, and I'm going to talk about trends and future developments of the rotavirus vaccine. So, the World Health Organization recommended the implementation of a rotavirus vaccine in 2009, and it was recommended to be administered along with polio, diphtheria, and pneumococcal vaccines in infants. And since then, there have been substantial declines in global severe rotavirus cases. And in the US, one study showed that positive rotavirus tests declined by 86% in children less than 5, only 4 years after the rotavirus introduction. As shown in the graph, the World Health Organization estimates that globally, 59% of infants have received their final dose of the rotavirus vaccine as of 2025. And then future developments of the rotavirus vaccine. Most of the vaccines in development differ in their routes of administration, and they're believed to improve protection in infants. So there are 2 new neonatal vaccines that are currently in development, and it is believed that vaccination during the neonatal period could provide additional protection during the first 2 months of life. And also, due to the administration route avoiding the gut, this vaccine may be less likely to cause intussusception. However, there is a concern about triggering an immature immune system, and the efficacy is in question due to the presence of maternal antibodies against the rotavirus. And then another type of vaccine in development are non-replicating injectable vaccines. And injectable vaccines are thought to bypass the issues of oral vaccines, which could be poor absorption or gut interference. And also, since it's injectable, this would also decrease the risk of the intussusception. However, the efficacy is in question of these vaccines also. One reason is that having a non-replicating or non-living vaccine may not trigger a proper immune response. And also, since the administration bypasses the mucosal barrier, it might not properly trigger the immune response that reaches the gut, which is where the rotavirus replicates. These are our references. Thank you.